we have to select. Threshold calculation for SFW, 5 points, 50 plants, and from there we can have to calculate the percentage, like 12%. Here 50 plants, 6 infested for SFW, so it is 12%. Here these 50 plants, 3 plants for IW or a larger worm, so it is 6% infestation. During reproductive, one more things we have to see, that is the infested plant, that means the cob infestation. Here you see there's 50 plants, 8 infested, so it is 16% infested. How we will put this data? So this will have to format like this. Like in Los Banos Laguna, we have actually maize planted on 15th February, this last 15th February. So within 15 days, we have to start our monitoring and especially scouting. Maybe on 15 this March too, first this scouting we will done. That is at 15 days, the growth stage, maybe D1 or 2. And the window panes, SFW, maybe 6%, IW 24%, IW maybe 0, and infested plant maybe 0. After 21 days, again we will go, maybe on
Dr. Alam,
days,
we need to wrap up, please.
So I'm just finishing because I have lots of 4 things. So that's why 3 more slides. Okay, please. So these have to 5 steps we have to repeat. That's the regional moth count before going to the field with the report. And for before field, we have to pheromone traps. 1, this male insect per trap per day is the threshold. Scouting, as I already told you. And based on this, we have to see. These are all the threshold limits, especially seedling. It must be more than 20%, SFW, IW. Mature, it is 50% because during that time leaves coarser. But in the reproductive, tassel stage is the more and more this dangerous. I'm telling you, this tassel stage. During that time, if the warm and dry weather, the control measure must be undertaken with most effective insecticide, pesticides, because during that time, if the infestation is high, the cob infestation will also be high later on. So last, the conclusion, I have already, it's an American pest now becoming almost a global pest, tropical, subtropical weather. It's very dangerous due to its highly destructive, especially migratory behavior, wide host range, large environmental and habitat suitability, high reproductive rates. Effective timely management is very, very much essential for this with monitoring and scouting based. Few words about effective management. Don't rely on chemical pesticides because there are lots of biocontrol agents. So we must have to go for integrated pest management where with biopesticides and host plant resistance and agroecological management, which is actually following by these talks. So I am acknowledging these institutes and also Dr. McGrath. And thank you. I'm just ready for your questions and now and also at any time. Thank you very much.
Thank you very much, Dr. Alam, for your presentation. As mentioned, feel free to type in your questions in our Zoom Q&A section. Okay, now we proceed to our next resource speaker. Allow me to briefly introduce him. We have with us Dr. Sevgan Subramanian. He is a principal scientist at the International Center of Insect Physiology and Ecology. He has over 19 years of research for development experience in Africa, and his significant research contributions are in the development of IPM strategies for horticultural and staple food crops, biopesticide research and development, climate change impacts and adaptation, and edible insects. Ladies and gentlemen, our second resource speaker, Dr. Subramanian Sevgan. Dr. Subi?
Subi, please put on your video.
Dr. Subi, you might be on mute.
Subi, you have to unmute and switch on your video.
Unmute myself and I'm switching on my video.
Yes.
And are you able to see my presentation now?
Yes, sir. You can try it on full screen, please. There you go. All good now, sir.
Yeah.
Thank you.
Okay. Thank you. First of all, thanks to Dr. Prasanna and also to our colleague from SIACA for giving us this opportunity for making this presentation. I'll be talking about one of those IPM techniques which Dr. Alam presented previously, biopesticides for fallow management. And this presentation, most of the work that has been done is done from my side and also from my able colleague, Dr. Kombi Akutse. Just to go before a quick introduction about SAP, we are based in Africa and we are a center of excellence in Africa for insect science with over 50 years of history, with more than 571 staffs working with us. And we do a lot of capacity building for African scientists, 150 to 180 graduates trained annually, and we collaborate with more than 300 partners worldwide. We recently had our new vision and strategy brought up, and our vision is to transform livelihoods through insect science and innovations. And our mission is to deliver innovative, nature-positive insect science solutions for people and planet. I would appreciate if you have time, you can have a look at the vision to have an understanding of where SAP is looking forward to the space of insect science. Before quickly moving on to the topic, I guess Dr. Alam gave a very clear, my thought process in terms of presenting this slide was to actually highlight the key points of intervention that we need to do for fallow management, especially the adults, which has the capability to migrate a lot. So they have to be targeted to prevent its migration. The egg stage, large number of eggs laid by the adults, and these egg stages have to be targeted by sustainable interventions. And also the larvae, which are the actual damage causing stage of this insect. And we need to work with the challenges that are there in terms of their concealing during the day, the economic damage that they have, and also the resistance to pesticides that these pest has got. The last stage, which normally does not take a lot of interest in terms of management widely, is the pupae, which is hidden in the soil. So we should also come up with strategies for managing this stage. So the whole idea is our management interventions should be holistic on all the different life stages to make sure that the efficacy is quite good. My talk will just not be restricted to the biopesticides, but I would like to expand it slightly to a broader scope of biorational pesticides, where the EPA definition clearly says biopesticides are certain types of pesticides derived from natural materials, animals, plants, bacteria, and other minerals. So within that scope, we have a lot of different things like the microbial pesticides, the plant-derived pesticides, and others, which I'll be taking you through with some of our work, largely from Africa, which is also, you can relate it to the Asian conditions as I speak. Why biopesticides? When you consider a parasitoid, it targets the egg stage and the larval stage. And when you consider host plant resistance, the target is mostly the larvae, to some extent on the eggs. But biopesticides has the potential to target all the different life stages which I mentioned. So there are fungus which you can target, which you can use to target the egg stage, larval stage, pupae, and the adults, all the 4 stages. The viral diseases targets mainly the young larval stages. There are recent reports on also its efficacy on some of the egg stages. And then we have the Bacillus thuringiensis, the BT, which targets mainly the larval stage there. You'll have this detailed information as I go through the presentation. So the idea is biopesticides are capable of targeting different life stages. So it has the potential to give a holistic control of fallow. When we start with the entomopathogenic fungus, these fungus are largely free living, and they have the potential to target the different life stages, as I said. They can be used both as augmentative control and also as conservation biology control efforts. Augmentative in the sense repeated application, while conservation in the sense conserving the ecosystem to have these entomopathogens work effectively to create epizootics. More recently, the role of entomopathogenic fungus, which normally also resides as colonizes plants as endophytes, is emerging. These are natural regulators of arthropods, and also as endophytes, they are non-pathogenic in most cases, and they also induce systemic resistance, which I'll be mentioning as I go forward. Yeah. Among the entomopathogenic fungus, there are two major groups of fungus which has been largely looked at in terms of management of lepidopteran pest, white muscardine disease, Beauveria bassana, which is effective against a different range of insects, termites, thrips, whiteflies, aphids, beetles, and all. With the Falamium also, the Beauveria bassana has entomopathogenic effect, especially against the adult more prominently as compared to the larvae and the eggs. The critical feature of this is in the early stages of its growth, the fungus will be pure white in color, which can be an indication that yes, you are having a Beauveria infection. The next one, which has largely been tried out in Falamium management, is the green muscardine disease, or the Metridium anomarea, which we call it so. Within this, Metridium anisophilae and also Metridium bruneum and Metridium rilli are ones that are now increasingly being seen on Falamium controlled space. So as with the Beauveria bassana, it is also widely seen in the nature, and it also has a lot of different hosts apart from Falamium as well. One of the difference between Beauveria and Metridium is Beauveria is more reported as endophytes, and it's more Hello? Light break. Hello?
Subbi, we can't hear you.
Okay. There is a slight power drop. So
Okay.
are you able to hear me now?
Now we can hear.
So Beauveria is more known for its endophytic properties, while Metridium is not so much endophytic in nature. So these are some of our findings when we tried out the EPFs against grown-up larvae, Falamium. We didn't find a lot of promising efficacy, while the Falamium are highly susceptible at the very young stage of egg and newly emerged larval stages. And at TCP, we maintain a lot of different isolates of entomopathogenic fungus, both Beauveria and Metridium. And of this, we have found some interesting isolates which have already progressed into commercialization, which I'll be presenting. Yeah. These isolates, especially the ECP7 and 78, which showed promise against Falamium control, has now been tested widely in the field in East Africa. And we have also tested the efficacy of some of these isolates on the adults. Why adults? Because adults are capable of by targeting adults, you can reduce the fecundity of the adults, and also you can use the adults as a kind of a vector for the entomopathogenic fungus. So as I said, in the adult stages, Beauveria bassana has a good efficacy comparable with Metridium and other isolates that we have studied so far. Whenever we apply biopesticides, I think we should make an effort to reduce the cost of use of the biopesticides. And in this space, innovative application strategies can help a lot. Horizontal transmission of EPF inoculums from one moth to another during the mating process can be quite useful. And in this regard, we have studied how the fungus, when applied on a male, gets transferred to the female, or from the female to the male, and what kind of mortality is caused. So clearly, both as the male and the female as a donor, the recipient sex tends to have a good level of mortality. And the additional advantage that you have is when a female is inoculated with the fungus, either from another female or from another male, their ability to start laying extensively decreases quite drastically. So this reduces the fecundity. We tried also compatibility of entomopathogenic fungus with pheromones, and we are able to clearly find that yes, the entomopathogenic fungus are compatible with the pheromones. So this gives a clear opportunity of developing auto dissemination tools, where the tools can be used for both monitoring and also for as a focal point for disseminating entomopathogenic fungus into the fallow mealworm population. This is what I was saying, that apart from spraying the entomopathogenic fungus, you can also try developing them as endophytes, clear endophytes, because the advantage of this is you have a very reduced cost of application of the fungus. Oops. Yeah. We have tried several endophytes, Trichoderma species Atrovidae herzianum, Hypocreolixi, Trichoderma asperellum, and others. And clearly, the number of egg masses laid when the plant is colonized with an endophyte decreases, and also the feeding potential also changes for fallow mealworm in an endophyte colonized plants. Are you able to hear me? Hello?
Yes, Dr. Subi, we can still hear you.
Yeah, thank you. Apart from the biopesticide being effective to the fallow mealworm, they should also be safe to the other biocontrol efforts that we have, especially the parasitoids. And in this regard, we have tested the safety of entomopathogenic fungus to parasitoids. And here is an example of Telenomus remus. And what we observed is Telenomus remus adults are not so infective to the entomopathogenic fungus. And also, when the parasitoid is allowed to parasitize an egg, and the entomopathogenic fungus is applied later, the emergence rates of the parasitoids are not very significantly impacted. So clearly, these two techniques could be integrated in an integrated pest management strategy for fallow mealworm. Following all these results, it is critical that it shouldn't remain in the lab. And in this regard, we have engaged with the regulatory authorities in East Africa right from 2019. And currently, the entomopathogenic fungus, both SCP-78 and 7, are registered in East Africa in 3 different countries: Tanzania, Kenya, and Uganda. And very recently, we also got it registered in Malawi for use in fallow mealworm management in these countries. The next biopesticide that has taken a lot of space in terms of fallow mealworm management is the nucleopolyhedrosis viruses, previously which was called as nucleopolyhedrosis virus. It's an obligate pathogen of Lepidopteran, so it can only survive on the host. The infection is through oral feeding, and early instas are very susceptible. Interesting enough, with Spodoptera frugipeda, these baculoviruses are highly specific. So normally in other situations, a baculovirus that is infective to one species will not be infective to the other species of the same genus. But interestingly enough, what we are observing from what literature has come out is it's not just Spodoptera frugipeda nucleopolyhedroviruses that are infective to fallow mealworm. Some strains of Spodoptera litura in Asia and some strains of Spodoptera litoralis in Africa are also capable of infecting Spodoptera frugipeda. This is because also the diversity of the virus that is found in the ecosystem. These are some of the products that are already available, and also some of them which has been tested in Africa: Foligen and also Litovir. Litovir is actually a Spodoptera litoralis virus which is infective against fallow mealworm, and Foligen is a fallow mealworm baculovirus. Clearly, the insect has to consume the virus, and it has a specific incubation period, so it is not very rapid in its kill. But the advantage with these viruses are they get transmitted through generations. So there is a kind of a horizontal and vertical transmission, so can contribute to decrease in population over time in the field. In Africa, there is some interesting work done by our partners CABI with the national partners in testing the existing baculoviruses for fallow mealworm control, and also looking at opportunities to see how recycled virus. So for instance, you spray a farm and you also get larvae from those farms which are infected with the virus, and you recycle this virus and try to spray, because mass production of this virus from commercial scale is costly. So this was tested in with the smallholders. The recycled virus are not as effective as the commercial ones, but still they are at times better than the untreated control, which can add to the foliar control strategies. The next group of biopesticides is the entomopathogenic nematodes. Clearly, the mode of action is they can infect through the spiracles and also the openings of the
To
insect.
be last
And this-
2 minutes, please.
Quickly, I'll finish up. Yeah. So there are several nematodes that has been screened. Clearly, commercial nematodes, Styracosa carpocraspae, gives up to 100% mortality, while there are also other nematodes that has been observed in the wild, which needs to be developed in terms of production and formulation and this. Yeah. The advantage of the nematodes, it's not just infective to the larvae. Very recent information from India indicates that the nematodes can also be infective to the egg stage and also to the pupal stage, giving a broader scope for control of foliar with entomopathogenic nematodes. The last one is the Bacillus thuringiensis, gram-positive bacteria, which infects the gut and makes the insect not to consume a lot. There are several commercial products that are available for BT, but more and more BT is looked at as a transgenic crop rather than the product to control pest, which I think Dr. Prasanna will cover in his talk. Apart from the biopesticides, there are several botanical options that has been tested all over the world. It is unique to the context of the situation. Botanicals present in the local situations contributes to foliar management. So a resource poor farmer can access some of these botanicals and also add to his repertoire in terms of foliar management. This is a study that was done in Ethiopia where we found that neem and also Schinus mole and other botanicals can contribute to foliar management or reduction in the larvae. The last one which I want to cover is biopesticide is not just about spraying a molecule for killing the insect. This is a traditional practice in Malawi where fish soup was said to be effective against foliar. The soup is not toxic to the insect, but it recruits a whole lot of natural enemies into the system. And this is what we studied. And these natural enemies, predators, actually contributes to the biocontrol effort of foliar. Yeah. So some of the advantages of biopesticides: it's ecologically safe, biodegradable, no harmful residues in the crop, often highly effective for management of pesticide-resistant insects such as foliar, and target-specific and safe to natural enemies. Can be effectively integrated, but the challenges with the biopesticides are costly in comparison to pesticides. So we need to have improved and local production and formulation techniques. Another way of reducing the cost could be innovative application strategies, auto dissemination, and others. Weak regulatory frameworks is also a challenge. We need to create enabling policies to register these biopesticides quite quickly in the different parts of the world. So in this regard, we have engaged in a lot of global level trainings on biopesticide production and also biological control agent productions. And as my previous colleague clearly highlighted, biopesticides are just a part of the entire spectrum, where active monitoring, use of biopesticide resistant cultivars, which you'll be hearing from Dr. Prasanna, and also capacity building are critical in establishing a comprehensive package of practice for foliar control. With that, I would like to conclude and thank some of our donors who have supported our work, and I'm happy to take any questions as it comes. Thank you.
Thank you very much, Dr. Suvi, for the presentation. We now move on to our next resource speaker. We have with us Dr. Samira Abulghasi Mohammed, a Sudanese national. She is an agricultural entomologist and biological control specialist. Currently, she is a principal scientist at the head of the plant health team at the International Center of Insect Physiology and Ecology. Her main area of interest are pre- and post-harvest management of native and invasive pests using agroecological based approach with main focus on biological control using parasitoid. Ladies and gentlemen, our next speaker, Dr. Samira Mohammed.
Samira, are you there?
Dr. Samira,
Samira?
would you like to start sharing your screen?
Samira, please share your screen. Okay.
Hi, Prasanna. Okay,
uh,
we are trying to share, but we have also a technical problem. I think you can proceed with other presenters. I had a technical problem from this other end. Please continue other presenters. I will come last.
okay, fine. So in that case, Sunny, I will go ahead.
All right. All right, sir. So we'll proceed with the next presentation. We have Dr. Prasanna again. Allow me to briefly introduce him. Dr. Prasanna is CIMMYT's Distinguished Scientist and Regional Director for Asia and Managing Director of the Berlog Institute for South Asia. He served as Director of the CIMMYT Global Maize Program from 2010 to 2024 and is internationally renowned for his contributions to the maize genetics, breeding, and deployment of hundreds of high-yielding, climate-resilient, and nutritionally enriched maize varieties for the rain-fed, stress-prone tropical environments in sub-Saharan Africa, South Asia, and Latin America through extensive partnerships. Ladies and gentlemen, our speaker, we have Dr. B.M. Prasanna. Sir?
Thank you very much, Sunny, and to the entire CRCA team for this wonderful opportunity to provide the perspective on various eco-friendly approaches for fall armyworm management. What I will be presenting is on host plant resistance to fall armyworm, for which CIMMYT is highly renowned. The breeding for native genetic resistance to fall armyworm started way back in 1990s when Dr. John Mim, this gentleman here, a maize entomologist, started screening thousands of land races and breeding materials for their resistance to various insect pests. And through that work in 1990s, Mim and team identified that there are two most important sources for insect pest resistance in maize. One is the Cuban flints. Another is Tuxpeno, which is a kind of a landrace accession in Mexico. So there are two excellent publications that followed in 1997, the insect-resistant maize, recent advances and utilization, where the protocols for mass rearing of fall armyworm, the sources for native genetic resistance to fall armyworm have been highlighted. But when you look at breeding for native genetic resistance to insect pests, be it fall armyworm or any other insect pest, it requires good knowledge of the biology of the host as well as the pest. The effectiveness here undoubtedly depends upon what kind of diversity of germplasm that institution has. Does the institution have an efficient insect rearing technique? Do you have an efficient artificial infestation protocol of the host so that you can generate robust responses, and what kind of breeding technologies or methods that the institution is following to transfer the resistance once it is identified in any breeding material into elite genetic materials. When the fall armyworm broke up in 2016 in Africa, CIMMYT, at the time I was the maize program director, we established this world's largest facility for fall armyworm screening at our research station called Kibako, which is the 2.5 hours drive from Nairobi. At this site, you can see 16 to such screen houses, each screen house 1/3 of the size of an acre. And it can hold several, you can grow several thousand entries within that particular screen house, artificially infest each and every plant, and not only at the vegetative stage, but also at the early reproductive stage, and then identify materials that are native genetic resistant. So using MIMS materials as well as many others that are adapted to Africa, we started screening germplasm at our Kibako facilities. Over 20,000 germplasm entries were screened against fall armyworm under artificial infestation at the Kibako facility. We also established a similar facility in Hyderabad, not to the same number of screen houses, but about 8 screen houses were there.